We're going to talk about these our own things, particularly in the CPF system which we are in. First, let us discuss something about the genome editing. What do you mean by genome editing? That's what first. When I ask what is genome editing, so when can anybody tell what is editing? What do you mean by editing? You want to, so suppose you have written an article, right? You have written one article, and after writing article, you must have given this your article to your friends. Kindly please check my article whether there are some mistakes or all. Your friends will take and he will do some editing. He will make some correction. If there are some mistakes, he will replace your, either there may be grammatical mistakes or there may be spelling mistakes. He will correct. That is editing, right? Similarly, now during evolution, the cells also do some mistakes. There may be some mistakes in the genes or in the genome. So now it is possible that we can now very precisely go to those mistakes done by the cell and we can rectify those mistakes in the genome. That is why, and for the prerequisite of genome editing, when you want to take up any project or any assignment or any research work on genome editing, you should have to have the crop plant which you are going to select. It should be whole genome sequencing should be available to you. And unless you have a whole genome sequencing is not available, you cannot edit. You cannot edit. You can, until unless you have the whole genome sequence. So one of the prerequisites for any genome editing says that the whole genome sequencing is must. Then only you can do the editing. You can go to the whole genome and then wherever the mistakes, you can find it out those mistakes and we can correct those mistakes. So whole genome sequencing, and for genome editing, one of the very important technology or the system has been developed, that is the Crispr CPF1 or your Cas9. So what is a Crispr? It is a CRISPR, it is a adaptive immune system developed by a bacteria. Okay? It is actually immune bacterial system, they have an adaptive immune system. You know that the bacteria, they are infected by some viruses which are called bacteriophages. Okay? Bacteriophages generally they infect the bacteria. So these bacteria, whenever the virus is going to attack, bacteriophage will infect, he will try to make what? Once a virus is attacked, bacteriophage is attacked, so he will inject his own genomic DNA. Okay, and then after the phages, so what the bacteria they do, they generally take, they copy some of the phage genes, they copy in your own CRISPR locus. This CRISPR locus is present within your bacteria. So suppose this is number one virus has attacked, he has copied this phage one your copy, and whenever there is another bacteriophage will attack, he will again copy another gene from the bacteriophage and sorry. Let me switch off this, otherwise it will go on disturbing. Okay, so there are a number of the bacteriophages may attack a single bacteria. So what he will do, whenever one virus is attacked, he will copy his genome, a small sequence of the bacteriophage, he will copy there, and whenever the next virus will attack, he will copy in this. Third virus is attack this. So he has copied 3 or 4 viruses which are attacked, number 1, number 2, number 3, different color. And then this CRISPR locus is transcribed. Once it's transcribed, it will produce the CRISPR RNAs. So whenever the next time, when the same bacteriophage is going to attack number one, this is number one bacteriophage, when it's going to attack in the second time, again in the same virus, so it has already the CRISPR RNA, and it will go and bind to that particular bacteriophage DNA, and it will degrade its DNA. So this is a kind of immune system the bacteria they have. And this system now we are using for knocking out a gene, or we want to modify a gene. This is a very good system the bacteria have, and now we are exploiting, or the scientists have used this system to knock out a gene, or we want to knock out a particular non-function gene, or we want to replace a very precisely a new gene, or we want to modify one or two nucleotides. That is all possible with this CRISPR. So let me go back to, so CRISPR is a family of the DNA sequence in bacteria. As I said, it was discovered in the bacteria, and the sequences of the CRISPR sequences, they connect, or they contain a snippets of the DNA from the viruses that attack the bacteria. So they copy a small fragment of the bacteriophage and they copy in the CRISPR locus. So these snips are used by the bacterium to detect and destroy the DNA from the further attack by the similar virus. As I said, if the same virus, a particular bacteria has infected by number of bacteriophages, so he has already copied all the SNPs, the small genomes of the bacteriophages in the CRISPR locus. And then whenever the second time the same virus is going to attack bacteriophage, he is going to destroy that. And that way he has become resistant to that particular bacteriophage. Well, we'll come to this. So what is CRISPR now? So it is a clustered, regularly interspaced short palindromic repeats. So if you see, these are the different repeats, okay, and there are the spacers, okay, and these are all palindromic repeats, okay. So bacterial CRISPR, this is a kind of structure of a CRISPR locus, and this is the bacteriophage. As I said, so this is already mentioned. So along with the CRISPR, there is another very important motif of the CRISPR is the CRISPR-associated protein, that is the Cas. And this is a CRISPR-associated, it is endonuclease, which make a double-stranded break. So the CRISPR locus, the guide RNA, which the copy of the bacteriophage which has copied, it is linked with the, it is accompanied by the your endonuclease activity called the Cas, either the Cas9 or CPF1 of the same bacteria, and they will go and bind to the same the sequence which is recognized by the or copied by the bacteria. It will go and bind to that region, and it will make a double-stranded break. And whenever there is a double-stranded break, as we know that the cell will try to repair those breaks. When he try to repair those breaks, they are called error prone. That means there are some error will definitely be there. It will be never like a normal. So there will be some errors. Either there will be some insertion, or there will be some deletion of some nucleotides. Either insertion or deletion. So there will be, so it will be never like your like a normal. So it will be like an error prone. So whenever there is a double-stranded break, so this repair will make, and there may be, and there will be a addition. This nucleotide has changed. So it is normal, not like a normal. So there may be some mutation in this, which called insertion or deletions. So whenever there is a insertion, one or two nucleotide, if it is entered, you must have been knowing like the open reading frame, the ORF. The open reading frame or the shift mutation may occur. Frame shift mutation. You know what is frame shift mutation? So when there is an insertion of one or two nucleotide, what will happen? Triplet codon, which code for a particular amino acid, it will be shift. So instead of initially there was a lysine, now they may be encoded by arginine or phenylalanine or any other serine. So ultimately, when there is a change in the amino acid sequences, the structure of the protein will also change. The biological function of the protein will also change. Either you can, if there is some premature stop codon because of the insertion or deletion, there may be some premature stop codon. And if there is a premature stop codon arise, it will become completely dysfunctional, that we call knockout, deletion. It is no more functional. The gene is no more functional. So this is the some of the advantages of your. So naturally, what did happen? So as I said, along with when we say the CRISPR, very important is a protospacer adjacent motif. This is one of the most important. Your sequence, generally the Cas9 or the generally the Cas9, it is an NGG, or since we are using the CPF1, it is an NTT. Okay, so this is very important because the Cas9, it will recognize this palm sequence and it will go and bind to the, and as I said, this is the guide RNA, okay, and there is this particular nucleus or the endonucleus has the two important motifs like IUCD and HNH, and this will make a double-stranded break. So this palm sequence is very, very important because this palm sequence is involved in the making a complex of your endonucleus into the target sequence. So if there is a double-stranded break, so two kind of things may occur. There may be a non-homologous end joining, as I said. So the two ends, they will be done. The cell will try to repair the double-stranded break, and there will be some insertion or deletion. This is a kind of mutation may occur. Or we can go for homologous directed repair also, or the recombination also, or the homology dependent recombination. In that case, we can make the guide RNA flanked by the sequences flanking in both the regions. Okay, so if you can add the sequences which is already present in your target DNA sequence, now this sequence can be precisely, we can insert. So gene can be replaced or knockout or new substitution. So as we say in case of transgenic, when we infect with the agrobacterium, we don't know where the tDNA is going to integrate. It may integrate in any of the chromosome. Okay, but since we are doing we are inserting some important genes or a sequence of a gene through CRISPR-Cas9 or CPF1 system, then we know where my DNA is, where my gene is going to be integrated. Because the sequence, the guide RNA is already known, it is from the chromosome number 5. So the insertion you want to introduce, it will be particularly in the chromosome number 5 only. It is not going to be entered anywhere else. So this is very, very precise, and we know where my gene has been integrated, in which particular locus. That is also possible. Okay, that is the advantage of inserting a gene using the CRISPR. Okay, coming to now my part, which right now what we are doing in our lab. So we have taken up this particular project, genome editing of banana, particularly the leaf call, using the CRISPR CPF1 system. Okay, and main objective is to make it seedless, and we are going to knock out intermolecular gene. I will tell, as I said in the very beginning, that why we have taken up this project. This is a bean call, which is one of the very important indigenous variety of this region. And as I said, it contains more than 100 to 150 seeds in a particular fruit. And these are the two products the entrepreneurs have developed, Bhim Shakti and Bhim Beta. And because of these seeds, their processing, when they are making this product, 50% of the pulp, 50% of this pulp is lost due to the seeds. And that is why there is a long-standing demand to make this being called seedless. Can we make it seedless? And till date, there was no any systematic research has been undertaken to make it seedless. So, taking up this as a challenge, so when I start studying the whole seed biology of the bean call and banana and all, and then I come across with one of the very important research paper that is in the you must have seen, this is a sitafal of the sugar apple. It contains a lot of very, very black, very big seeds, right? And in the Thailand, this is a Thai seedless, and there is one variety they found there is no any seed. No seed, complete seedless, your custard, your sugar apple. When they try to know what is the reason behind why they become seedless, when they did the whole analysis, they found there is one due to spontaneous mutation. One of the very important gene called inner new outer gene, which is involved in the synthesis of two important integument layers during ovule development. And this particular gene is involved in the synthesis of two outer integument layers. And if this gene is, and since in this particular mutation, spontaneous mutation, this gene was deleted. This is natural, spontaneously. Nobody has done artificially. Spontaneously there was a deletion, and because of that, this becomes seedless. So they have done the same work in the Arabidopsis, and they found this is a normal. When they deleted this gene, so there is a defective. The two outer integument layer is very defective, and there is either there is no any seed development, or even if the seed, the percentage of seed production in the Arabidopsis is very, very low. Almost like more than 80% of the seeds is reduced when they knock out this particular inner new outer gene. And taking this as my proof of concept, I developed a project on Vimcol, and we did a lot of analysis. So since that time, the Musa Balbishana, the genome was not available. A draft sequence was available, which is not very good. So I took the sequence of Musa acuminata, AA genome and DB genome. Musa acuminata and Musa Balbishana. There are two wild type bananas. So since Musa acuminata genome was available, so when I researched this whole inner new outer gene in the Musa acuminata, we found that it is present in the chromosome number 5. So taking that into case, so this is the gene structure of the inner new outer gene of the Musa Balbishana. So we already cloned that gene from the Musa Balbishana, that is from the Vimcol. We isolated the gene, and then we cloned and we analyzed the genes, and we found there are about 7 exonic regions. You know what is introns and exons, right? Introns and exons, So there are 7. If you can see, this is the first exonic region. This is the second exonic region. This is the third exonic region, 4th, 5th, 6th, 7th. So 7 exonic region was present in our particular gene. And I just want to give you just a comparison between what is the difference between a Cas9 and CPF1. I'm using CPF1. People, other labs, they use Cas9. In case of Cas9, it is found in the bacteria called Streptococcus pyogenes, whereas the CPF1 is the Leptospiraceae bacteria. And the advantage, what is the difference between is that if you see the structure of the Cas9, they needed transactivating RNA along with the guide RNA, along with the Crispr RNA to become a complete guide RNA. Whereas here, we don't need any transactivating RNA. So the structure is much simpler in case of CPF1. And you will see this is the palm sequence for the NGG and NTT. So generally, as I said, the guide RNA generally they recognize, or the Cas9 generally recognize the palm sequence, that is the NGG, and which lead to the make the binding of the guide RNA to the target, your DNA, and it always make either just 3 or 4 nucleotides away from the palm sequence. They make a double-stranded break, and it is a blunt. Okay, they make a double-stranded break, and it will generally lead to a blunt end. Your ends. However, when we see here, this is the NTT, and it go away. Generally, the digestion or the break of the double-stranded DNA break occur at least 9 to 10 or 9 nucleotides away from the palm sequence. 9 nucleotides away, and generally it is a staggered end. It do not make a blunt end. Generally, since it is a staggered end, So it generally release overhangs, like your other restriction enzymes. So generally overhangs is generally released. So that is why it is, if you see this is structure, you need only 2 RNA, that is a Crispr RNA transactivating variant, only 1 RNA is required. Blunt end cuts, staggered end cuts, proximal to the very close to the palm sequence, which is distal from the recognized sequence, which is away from your palm sequence. And it is a G, that is the very G-rich palm sequence. This is the G-rich, and this is the T-rich. Okay. So once we isolated the gene, we cloned the gene, we'll sequence the gene. After doing all, then we need to now design the guide. Which particular sequence I will be using to make your single guide RNA. For designing any guide RNA for genome editing, I use very user-friendly benchlink.com. This is a very good software which you can use for designing your Crispr RNA or guide RNA. So as you see in the cis, I need a guide RNA base pair. Okay, this one. I have given around 23 nucleotides. My guide RNA sequence should be around 20 to 23. 20 is required for the Cas9. For the CPF1, you can go up to 23. So I have give the input of 23 nucleotides. And what is the genome I'm going to search? The genome which I'm going to search is the Musa accumulator, because I want to see, once you design a guide RNA, this guide RNA is a very 23 nucleotides. It can go and bind to anywhere of your whole genome. And wherever it will go knockout. It is going to what will happen? It is going to knock out, or there will be a lot of off-target effect. Some very important function genes can be deleted or mutated. And that is why we have to be very careful when we are going to design the guide RNA. So after doing this, it has shown me several guide RNA, but there is one guide RNA, target 1, showing 100% knockout with the inner no outer gene. And if you see, it has very low target effect, 0.1, very less. So we can take 100%, it is going to knock out the inner no outer gene of your Musa balbishana, but it is having only 0.1 to other sites. That means it has a very low off-target effect. Okay, and the target 2, the second, it is showing 50% your knockout of the Musa balbishana. Since there is a one only nucleotide difference, since I am using a Musa accumulator and I'm synthesizing the guide RNA for Musa balbishana, right? So there may be a one nucleotide difference between Musa accumulator and Musa balbishana. So I can ignore that also, because I don't have any other option, because the Musa balbishana genome was not available with me. So that is why I have to compromise with the single nucleotide difference, which is showing 50%, but you will see the other off-target effect again is showing very less, 0.1 to 0.2. So I can assume that, okay, we can expect that this is also going to be, maybe in my Musa balbishana, instead of T, there may be A. I don't know. We still don't. But now it is available, so we know now. So this is the whole which we cloned this gene. This is the same, okay, this is my name they have given, inner no outer gene, and guide RNA chromosome number 5 of Musa accumulator. This is a Musa balbishana, this is a Musa accumulator, and when we compare and we design number 1 guide RNA here, you will see ATT, this is the palm sequence. And whenever we design any guide RNA, we should not include the palm sequence in the guide RNA. It should be always away. You should not include the palm sequence when you are designing the guide RNA. So we have taken GGT-AAU up to here, and if you see GGG, this is another palm sequence. And this is my second, this is an exonic region 2, and this is exonic region 3. Okay, so 2 target sequence we have selected for synthesis of guide RNA. Okay, so this is target 1 and 2. And now, once we have selected the guide RNA, 2 guide RNA we have selected, now the thing came where we now to clone it. Right? We need to clone that. So we have developed, well, this work I have accomplished with the Washington State University, USA. I visited there, they invited me as a visiting scientist for 6 months to initiate the work on genome editing, because they have also initiated the work, and that time we also started just working. So since I have a very good relation with the professors or Professor Okita from Thomas W. Okita, he is a renowned biochemist and biotechnologist who is working on starch biosynthesis. So I have a very long association for last 20 years. We have a very good association with this Professor Okita. So he invited me as a visiting scientist, and then I got the opportunity to start working on genome editing. So there we have developed all these vector systems in Professor Okita's lab, okay, in the Washington State University. So one of the vector system which we are using is based on the Golden Gateway cloning system, where we are using not like a normal restriction enzyme, but here we are using type 2S restriction enzyme like BSA1. Okay, I will tell you, come to that point. So this is the one vector system, that's a PSH923B, which is, now this is a guide RNA cloning site. Okay, we can clone the guide RNA here, and this guide RNA cloning site, it is here, and it is flanked by 2 BSA1 restriction sites. Okay, and this is driven by the Oreja cycle O3 or U3 promoter from the Oreja cycle. That is a U3 promoter, and this is the same vector, but here instead of U3, it is a U6B2 Oreja sativa promoter. Only the promoters are different in both the vector system. Otherwise, both are almost same. So one we are using U3 Oreja sativa U3 promoter. Otherwise, you have Oreja sativa U6B2 promoter. Otherwise, both the vectors are very similar. Okay, and as I said, so this is some direct repeat, all 3 terminator. So this is the whole expression cassette, okay, which we are using. So this is another vector system I used, which is flanked by 2 same your Oreja sativa, your U3 and U6 promoter, but here it is containing some your viral or ribogenic motifs. One is containing your hammerhead ribogenic virus, okay. Other one is the hepatitis delta virus. So these 2 sequences, the purpose of including the 2 ribogenes motif of these 2 viruses is that because these sequences involved in the self-cleaving, because ultimately the guide RNA need to be cleaved. So these flanking regions of the ribogenes may help in the self-cleaving of the guide RNA. So that is the reason we flank here with the 2 sequences of your ribogene viruses. One is your hepatitis delta virus, and the other is the hammerhead virus. And finally, so these 2 are containing your expression vectors. These 4, whatever I have shown, these are the expression vectors, okay, where we can clone the guide RNA. But once we clone those guide RNA into those your expression vectors, then finally we need to clone the whole expression cassette into a binary vector. Because ultimately we need to transform this whole guide RNA. We need to introduce into the host plant, like in Musa Balbishan or in the bean corn. So the process of introduction, it should be either through particle bombardment method or through agrobacterium mediated. So since I choose to use the agrobacterium mediated genetic transformation system, so I need a binary vector. So this is a binary vector, okay, which is again having a, here in the binary vector, CPF1 endonuclease activity. Since this is quite around 15.4 site, Now, the earlier if you see the sequence, there is no any endonuclease gene. Because as I said, you need a guide RNA, along with that, you need a, which is going to cleave, which is going to break the DNA. It is the endonuclease, Cas9, or the CPF1. So that has to be cloned along with your guide RNA. So since these are the as I said, these are the expression cassettes. So here you can see this is the CPF1 endonuclease activity, which is driven by the UB quitting promoter. And it has a hygromycin as a plant selectable marker gene, kanamycin as a bacterial selectable marker gene, and your left border and right border. So if you see, this is the left border and this is the right border. Okay, so this is the tDNA. And we will clone our DNA here. Okay, there is a cloning region. This is the cloning region. The whole expression cassette, which we clone in PSH 923 or 924, we are going to now clone the whole expression cassette into this region. So this is a binary vector system. And once we develop these, all the binary vector systems, now we need to design a series of primers. These primers are very, very important for cloning your guide RNA. We need to clone a series of, yes, the number of primers need to be designed. Okay, and you will know why we have designed these primers. You will see these, all the primers when we are using it during the cloning. So what is the flow chart? So we have a guide RNA, we have designed a guide RNA, and it is already having overhang with the adapter. We need to use some adapters to clone our your guide RNA into the first PSH91 or expression vectors. Okay, and then we need to ligate this guide RNA into either 923 or 923B. This is the first clone we have got. Once we got the first clone, we will amplify this whole expression cassette. We will purify the product, and then finally we will clone into the PSH916. This is the binary vector. So this is my PCR product. This is the expression cassette, whole expression cassette, and again there is two restriction sites, BSA1. So when we design this whole vectors and your different sites of restriction there, we intended we have incorporated BSA1 site, because BSA1, as I said, it is used in the restriction of tool. As restriction engine we are using, it need to be cloned. So we have already cloned this BSA1. This is also having BSA1 here also having. So when we digested with the BSA1 916 and digested my PCR product with the BSA1, finally we got a digested product, and then we ligated. Finally we get the second clone. This is the guide RNA clone into the binary vector. And now we verify our guide RNA through sequencing. There may be chances of some mutations, so we need to verify. But once you complete the cloning, we need to verify, and then after verification, if you find your guide RNA is perfectly right, now this vector system is ready to introduce into the banana. Okay. So as I said, the golden gateway method we use. So generally, what is the BSA1? It is a 4 nucleotide, 1, 2, 3, 4, 5, 6. It is a recognized sequence is this one, and it generally digests. It is not like your type 2 other normal restriction engine. It will digest 1 nucleotide away from the recognition site, and that will lead to develop 2 overhangs. 4 nucleotide overhang will develop. So this is one restriction site of BSA1. This is another restriction site, you will see. And it is going to digest here, and here, here, and here. So this inside portion will be released. Okay, this fragment will be released, and so this fragment will be released, and this is the overhang. This is we have used as an adapter. Okay, so you know what is the adapters and linkers. You know, during cloning we use a lot of adapters and linkers. Your backgrounds, you're all MSc students, MSc, PhD. So I assume you know what is adapter, restriction engine, cloning, linkers. So here we have used the same sequence we have developed to clone our guide RNA with the same sequence of adapter. So example of DNA fragment which is having two sides. The addition with the BSN releases the central fragment. It will reach the side. Inside it will be released. And with the 4 nucleotide overhang, this is very, very important for designing our guide RNA. So the guide RNA which we have designed, we have to add these 2 overhang in order for the cloning. Okay. Now this is the flow chart. So this is my first guide RNA which I have designed the primer for the Crispr. This is in the exonic region 2. And first, after designing them, we need to mix them at a concentration of 2 millimole, both the primers, forward and reverse primer. We mix it in an equal 1:1 ratio, and then we just boil it for 10 minutes. After boiling, and then slowly we cool in the room temperature. So they are complementary here. So we can see both. This is become now the primer duplex. So it has developed a duplex primer. Right? Now this is my guide RNA, double-stranded guide RNA. And if you see, there is an overhang, AGAT. This is adapter. Here is an AAAA. Now, this is my PSS, as I said, PSS 923 or 924, where there is a 2 restriction site. This is your U3 promoter or U6 promoter, all 3 terminator, and UTR and other things are there. So, first thing is that we need to digest our plasmid DNA. This plasmid DNA we need to digest with the BSA1. So this is a 2 rigonous site of the BSA1. So when you digest, what will happen? This central portion will be released, right? When you are going to digest, this portion is going to release. And once this is released, these 2 BSA1 sites abolished, right? It has gone along with the fragment. There is no more any BSA1 site left over. And this is my guide RNA with the adapter. So you can see AGAT. AGAT. Is it okay? AGAT. It will complement it to T, C, T, A. Here is the AAA, TTT. Right? We have designed this, you can see AGAT, AAA. So it has AAA, AGAT, AGAT. This is not present in the guide RNA, mind it. We have added these 2 adapters in order to clone in the PSS 923, because this vector system, when we are going to digest with the BSA1, it is going to release 2 overhang. That is the TCTA, TTTTT. So I need to add the vectors, adapter, AGAT in order to complement this region, and AAA, another adapter, to ligate this region. Got it? So then we just add these 2 mixture, and then we use the polynucleotide kinase and the P4 DNA ligase, and it kept it in the overnight at 14 seconds. And then after next day, we transform the whole ligated mixture into the another competent cells like XL10 gold and plated on an LB ampicillin, because this PSS 923 or 924, it is having a ampicillin placed marker for bacterial selection. So we use it, ampicillin plate, and then after next day, we have got so many colonies. I got almost like more than 100 colonies in a single plate. So I picked up only 6 colonies, and then I isolated the genomic DNA. Okay, sorry, plasmid DNA. After isolating the plasmid DNA, and this is the lambda marker. Now, as I said, in the original vector system, if you see, this is the original vector system. Is this one. Inside, in the guide RNA clone site, there is one restriction enzyme, NCO1. Mind it. Remember that NCO1 is present in your original guide RNA cloning site. Okay, so when we are going to digest with this, when this region, here is there is one NCO1 site is also there, NCO1 restriction site. So when this region is going to abolish or replaced by guide RNA, the NCO1 site is also abolished, right? I don't know. NCO1 will also be released, right? It will be removed within this fragment. So when my guide RNA is going to replace this region, this NCO1 site will be abolished, right? So when this is abolished, so we digested my all plasmid DNA with the NCO1. So wherever there is a digestion, this is a digested. It means what? Still the original vector system. There is no any guide RNA. There is no guide, still NCO1 site is there. That means your guide RNA is not cloned in those colonies. But I was lucky to get at least 1, 2, 3, 4 colonies, plasmid DNA, abolish the NCO1 site because there is no any digestion. Right? Since there is no digestion, so plasmid DNA digests with NCO1 design. This is the undigested product, and this is for the control. Now we have to always run one positive control, one negative control. This is the same plasmid DNA, this original vector system that is with the NCO1, and then the 3, 4, 6, 7, 3, 4, 6, and 7 positive clones, and clone number 5 and 8 negative clones because they still having the NCO1 site. The original NCO1 site is still here. That means this fragment is not replaced by the guide RNA. So this is the way of confirmation. Which colonies or which particular clone you got a positive clone simply by digestion with the NCO1 site. Then, now we use again, these are the 2 different primers. Okay, what are the primers? So amplification of the whole expression cassette using U6B2 BSA1 forward and USABR. This is U6B2 forward primer base, and second one is the BSA1. Yeah, this one. So this and this, if you see, both containing one BSA site. We have designed the primer in such a way it contains a BSA1 restriction site, because ultimately the whole expression cassette I need to clone it to where? Into a binary vector, right? And I have to introduce the BSA1 site because in the 916 there is already BSA1 site is there in the cloning site, cloning region, in the MCS region. So the BSA1 site is there, and this BSA1 site is not present in this region. So when we are designing the primer, we design this primer in such a way that it includes one BSA1 site here. This is a BSA1 site. There is another BSA1 site here. We included, and then we amplified. So this is an amplified product. The whole expression factors, U3 promoter or U6 promoter with your guide RNA and Pol3 terminator and other things. Okay, the whole expression cassette we have amplified and then we purified. We combine all the PCR products since they are all are same. So we combine, and then finally we need to now clone back this whole expression cassette into 916. And then this is again in the BS 916. This is the expression cassette. Again, it is contained to BSA1 site, as I said. That's binary vector 916, which is containing your CPF1 endonuclease, hydromycin, selectable marker gene, and other things, which is containing your TDNA, left button, right button. So this is the cloning site of the 916. It is again flanked by 2 BSA1 site. And the PCR product, as I said, when we design the primer, it also contains the BSA1 site. So the PCR product, both the region, both the left and right, or the 3' and 5' contain the BSA1 site. So we digested the 916 with after digestion with the BSA1, and this my PCR product is also digested with the BSA1, and then we gel purified both the fragments, we ligated, and transform the ligated pearl, finally transform into E. coli. And now here we plate it on a kanamycin plate, because finally here 916 is containing a bacterial selectable marker gene, that is the kanamycin. And this is the finally, as I said, this is my vector. So finally we cloned the whole my RNA into this region, and finally we did the quality PCR. Simply we did the quality PCR, and we amplified the whole expression cassette of the quality PCR, and then we isolated these quality PCR, since they give us the right amplified product. So we isolated the genomic data from those plasmid clones, or these bacterial clones, or the plasmids we have isolated, and finally we sequenced them. Finally, we send it for sequencing for verification. So this is number 1 clone. We have cloned Crispr1. This is in the exonic region 2. And this is again with the U3 promoter, and this is with the U6 promoter. This is another CRISPR2 we have cloned with the U3 promoter, CRISPR number 2 in the 3rd exonic region. This is verified, and we compare the sequences. They are perfectly all right. So now my binary vector systems. Okay, so after verification, your sequence verification, well, this we have done almost like 4 or 5, and then as I said, we have also used another vector system which is flanked by hepatitis head virus and the hammer delta virus. Okay, so since these they are almost same like 923 and 924, but only flanked, the guide RNA region is flanked by the 2 ribogen of the virus sequence, which is we expect that once we use this, maybe it will help in the self-cleaning of the guide RNA. Here the slightly different system we have used. We designed this is the guide RNA. Okay, this is the guide RNA or guide RNA 1 we have designed, and the primer also we have designed in such a way that, because this is a guide RNA all, and G guide RNA 1 forward. This is a forward primer, this is a reverse primer, and when we did the whole PCR, so we got a PCR product of around this whole plasmid we have amplified, along with this is my guide RNA. So since my primer containing the guide RNA, this is my guide RNA, so the amplified product will include this guide RNA. So this, where initially there is no guide RNA, but here this PCR product or the amplified plasmid will contain your guide RNA. So this is again for the 924, we have done the same way. So the flow chart is like this. We have generated the PCR, generated plasmid subjected to, first we have amplified the PCR product, and then after the PCR you use the 2 primer, guide RNA and the reverse primer. When you amplify, you get a mixture of your parental plasmid and amplified plasmid, right? And the amplified plasmid containing your guide RNA, whereas your parental plasmid DNA system, it do not contain your guide RNA sequence. Now we need to remove the parental your vectors. So we generally digested the whole PCR product, we digested the DPA1 restriction in there. Now DPA1, the purpose of digesting your with the DPA1 is that DPA1 only recognize the methylated recognized sequence. So the parental plasmid, the DPA1 side, it is methylated, whereas the amplified your fragment of the same plasmid, the DPA1 restriction side is non-methylated. So wherever the parental plasmid DNA, we want to remove it because it do not contain the guide RNA. So we need to cleave those. So we digest with the DPA1. So DPA1 will recognize only the methylated DPA1 side, and it will chop. So your parental plasmid DNA will be chopped, it will be degraded. Here, your plasmid DNA containing your guide RNA will be remain intact because DP1 cannot digest the non-methylated restriction site. So that is the purpose of digesting with the DPN1. Then with the whole PCR product, we have just go for ligation, transform into XL10 gold, and then we did the digestion again like NCO1. It is also having the SAC1 site in the. If you see there is a instead of a NCO1 in the earlier vector, here they are having SAC1 and SAC2 sites. So if your DNA is cloned here, if your plasmid DNA is cloned here, this site will be abolished, right? Because it is not going to amplify. I'm using the primer here and here. This is abolished. This fragment will be removed, and ultimately you will have those clones which cannot be digested. That means if there is no any SAC1 site, it's removed, right? So the plasmid DNA which cannot be digested with the SAC1, they are your positive clones, and those which are digested with the SAC1 like this, they are your negative clones. And finally, we combine all those, and then finally we cloned it into the 923. All those clones we have taken, and finally again we amplified the whole expression cassette. So this is the expression cassette. Again we cloned it, we amplified, and finally cloned into 916. Okay, so that way this is again we have to verify through sequencing. So we have verified guide RNA1, that is one clone, and this is another clone, guide RNA2, Crispr in the exonic region 2 and exonic region 3. Finally, once we verified all the guide RNAs which we cloned into the 916, both containing your flanked by the virus over or the ribozymes as well as non-ribozyme, we had 93 or 96. So we have developed a series of vector systems. Okay, this is the PSH 9101 containing the Crispr1 driven by U3 promoter. SS2, this is 916, same Crispr2, U3 promoter, Crispr1 with the V2 promoter, base promoter. Well, this is another I used since I was working with the Washington State University. So I also involved in the starch biosynthesis. So I cloned guide RNA for them also. This is another for the rice, for the chlorophyll oxalase enzyme. This is another chapter. Well, these are all with the containing the ribonucleic. Okay, these 4 vectors containing 9314, which is having the hepatitis virus as well as the hammer delta virus, and these vectors do not contain any. Your guide RNA do not flank by any virus variants or virus regions. Okay. Well, as I said, simply preparing a guide RNA, cloning, not going to serve. Whatever you have now developed, these vectors after a series of experiments, you have developed so many vectors. Now your final is to introduce. You need to introduce into the banana through agrobacterium, right? So first job is that we need to develop a very, very reliable, reproducible in vitro regeneration system in the being called the Reservoir Division. So we have already developed a very good, very reliable in vitro regeneration system in being called. We generally take the male flower, okay? You know, Colville male flower. So we generally remove all the outer brackets. We go up to the 11th, from 11th to 8th, or sorry, from 7 to 5, 7 onwards to up to 1 or 2, from 7 onwards. We take those very mature male flowers, we culture on a callus induction medium, and these after almost like 3 to 4 months, they develop some very good embryogenic color. Well, the frequency of induction of embryogenic calli is very, very low in vitro as compared to the other cavendish banana, because it is a BB genome. The genotype is different as compared to the other cavendish banana. They are triploid, AAB or BBA or AA. But here this is a BB genome, it's a diploid. So the frequency of getting this kind of embryogenic calli is very, very different. Although we have got, but the frequency is very low. No problem whatever the embryogenic calli you have got. Then we very carefully we take these embryogenic calli and then grow them in the liquid medium to develop the embryogenic cell suspension. Okay, so we develop the very nice, beautiful embryogenic cell suspension, and if you see, this is a beautiful picture of the somatic embryos. Okay, this is a very good small pictures of somatic embryos we have developed in our lab, and this is all our lab experiments. And then once you develop very good ECS, that is embryogenic cell suspension, then we need to mature them. Say for any embryo, okay, how a somatic embryo is different from a genetic embryo. How a embryo is developed in the seed? By fertilization of what? Male gametophytes, female gametophytes. They are all haploids. When they fuse, you get a zygote, single cell zygote, diploid. And when this zygote undergo what? Millions of matrix division to develop a mature embryo. Until unless your embryo is mature, embryo do not germinate. Even if it is germinated, this is called precocious germination. You know what is precocious germination? That is, if an immature embryo will germinate, it will display only those organs which are expressed in those embryos. Either the leaves are not very well developed, or the stem will be very weak, or they may not develop proper root. So those seedlings cannot survive. Precocious germinated seedlings do not survive in the nature. They die. So you need a very robust, mature embryo. When it germinates, it's going to give you a robust seedling. Similarly, in case of your somatic embryogenesis, since we are using here your male flowers, so these are the somatic embryos. They need to be also mature. If you allow them to germinate, these embryos immediately, if you allow them to germinate, they will produce a very, very weak seedling. So we need to mature them, and that is why we need to put them in another medium, now in a solidified plate on another medium. And if you can see these small granules, they are beautiful somatic embryos, mature somatic embryos. Almost from here up to here, it took almost like more than 2 years. The duration. It took almost like more than 2 years from this stage to this stage. And once we get a very good mature embryos, after 2 months of culture, then we put them in the germination. In another medium, where now we allow our somatic embryos to germinate. So within 2 weeks, it starts germinating. You can see these are turning very beautifully green. This bulge out, the somatic embryo will generally bulge out, and there will be a chlorophyll synthesis, deposition of chlorophyll synthesis, and there will be a lot of starch deposition. The color will be slightly changed, they will completely become white, because you know that germination is a very, very energy consuming process. So those somatic embryos which will start, they want to germinate, they generally deposit a lot of starch. So that is why they become completely whitish color. So when your callus will be a whitish color, or somatic embryo will be very, very whitish color, that means they are now ready to germinate. Once they become whitish, they will start synthesizing the chlorophyll. And once the chlorophyll is synthesized, that is called the nodules or the growing centers. That is the region where there is a localized cambium-like cells will develop. And this is the region from where you will get a shoot. Okay, so we get a shoot from here. You can see this tiny shoots, and then once you get these small tiny shoots, we immediately transfer them to another medium, and you can see here we get a lot of shoots. Okay, we exclude all those shoots. We can see all these somatic embryos germinating. Then we put them in a further development in a shoot multiplication medium, and then finally rooting medium. Okay, once they rooted, then we transfer these plants to the honey. So this system you need to develop before adopting any genome editing or transgenic, any kind of your work or genome engineering as a whole. If you want to start the crop you are going to work on, that this system should be in your hand. If you do not have this system, forget it, your genome editing, forget your transgenic. You are not going to. Until a very robust in vitro regeneration tissue culture protocol has to be there in your hand. Until you have that, you are not going to achieve anything. And then now we have actually, this is our original experiment. Then this is, we have infected our, this is the ECS, infected with our ECS of bean code, we infected with our PSS02. And sorry, is it Lima, is it 02 or 03? 02. So this is a 02, PSS02 having my CRISPR exonic region 2, right? This is exonic region 2 with the U3 and U6 promoters. Okay. So this is now we infected our ECS with the PSS guide RNA binary vector. Then this is under the selection, and this is under selection. After selection, we put them in the germination, and you can see now we have got this germination because now our protocol system was ready. That is why now we have immediately started infecting our ECS with your guide RNA binary vector containing your guide RNA. And now you can see this is now start germinating. So we again transfer those small somatic embryos into further for fresh plate. So you can see now beautiful germination. And this is finally, you can see some are dying because still we are selecting in the hygromycin. So those which may be escape, there may be some escape, false shoot, may develop resistance against hygromycin. Then we need to eliminate them in the subsequent. So there will be elimination of those. You can see these are still dying, and however, we are getting some good shoots which are very healthy. And finally, we transfer them in the rooting medium. Right now, these plants are in the medium. Okay, we have got around around 50? Yeah, around 40 to 50 plants right now in the rooting stage, or some are in the proliferation stage. And if once we get these plants, we are now going to sequence. This is not the imaging sequence. This is only prediction, but we are going to expect. So this is my enogene suppose. This is the enogene target sequence. So I'm expecting all these transgenic lines, sorry, the CRISPR edited lines maybe undergo some mutation. There may be 7 mutation, 73, 75, very good deletion. And when there is a change, there are some deletions. What will happen? The whole ORF, the open ended frame will be changed. There may be some premature codon, what we are expecting. This is whatever we are working as a proof of concept. Nobody has done on gene in the world. Nobody has worked. We are the first person who are working on the beam code. So we don't know what kind of mutation we are going to get. We don't know. Until unless we sequence our CRISPR-edited plants and finally we get the sequence. And then we can identify what kind of index, whether there will be a deletion or there may be some insertion. Whether insertion or deletion, ultimately our objective is to mutate that. My objective is to mutate the inner no outer gene of the beam code so that it enables to synthesize the 2 outer intervalent layer in the oval development. And that may lead to reduction in the seed. Instead of 150, if I can reduce up to 30 or 20 or 30 seeds also, that is also going to be a great help to the farmers or to these entrepreneurs. Right? If you can reduce the seeds, if you can make it complete seedless, well and good. Beautiful. But if still you can reduce to, even if you reduce 50 or 70% seeds, instead of 150, if you can come down to 30 or 25 or 30 seeds, that will also be a good help to the industry. So this product which we are working, it is a great boon to the industry. Thank you so much. Now you have any questions, please. I expect a lot of questions from you. Yeah. As I said, the lab in which I was working, they were using the CPA1 system, and as I said, this is much more simpler in the structure as compared to the Cas9. With the Cas9, you need 2 RNA, cross-activating RNA as well as the Crispr RNA to make a full guide RNA. Here is you need only Crispr RNA. You don't need any cross-activating RNA, and it will give you a staggered end. There is advantages over your Cas9, right? And moreover, the size of the CPA1 is smaller than the Cas9. So it will be easy for us to clone, as well as if you have a very large vector, it will be very difficult to transform into bacteria or transformation or handling with bacteria. So that was an advantage for me that the lab which I was working, they were using the CPA1, and I was fortunate to get CPA1 to use it. Yeah. So that. No. Whether you have understand or not, that is a thing. Because we must be come across this kind of cloning for the first time. Right? That also not the normal cloning, it's the gateway cloning system is slightly different from the normal cloning. We have to do a lot of manipulation. First thing, the development of vector was a big task for us. The vector system which we need to develop, because the restriction site we need to introduce and the adapters we need to introduce. That was a big challenge. So once we completed development of vectors, then our challenge was to design the guide RNA. And then how this guide RNA is going to be cloned. That was again big challenge, but for God's sake, it worked very fine. In a single goal, or in a single way, the one day only, in a single goal, we completed this goal. Although it is a very, very tough, sometimes we need to spend more than 14 or 15 hours in that time when you do this kind of research. So that time, we do not see that time. Okay, when you start working in this kind of area or in this kind of phase, we do not see the time. What is the time? Sometimes I still remember one day I was cloning, I was not getting the right clone properly. I was repeating, and in the morning I was seeing the sun has rised. And the whole night I was working. And I saw the sun start rising. I told him, so that kind of, you need that kind of your motivation also to work on these kind of aspects. When you people have a very good potential, you can definitely do some advanced research. Although our lab is, or our gold center, as well as the department, we do not do the basic research. We are doing very advanced research. That's all about it. Any more? Yeah. I don't know. Maybe. This was unpredictable. I don't know what kind of relation I'm going to get, because they are still yet to be sequenced. The molecular confirmation is required.
my question is then, is there any basis for this because of deletions and all?
No. There is no any limitation. There may be a 75, 60, or there may be one single nucleotide deletion. Maybe a single nucleotide. So this is a different kind of. So that is why for Crispr edited plants, you don't need one or two plants. You need this is a mutation. So you need a very good population, mutation population. You need at least 100-200 plants, or maybe more than 200 plants, to screen your mutants, where you are going to have the desired trait. Simply one nucleotide deletion may not affect the function of the gene. Still, you are getting a lot of seeds. So you need to find out what kind of deletion going to completely dysfunction the gene. It is not like a transgene. You have introduced a foreign gene. You got 10 plants, and one plant showing, okay, good, very good expression, single copy integration. You are showing very high expression, 100% success, or if it is a disease resistant, showing 100% disease resistant. If it is insect, 100% resistance against the pest. You got it. You need only one line. And that line, you give it to the breeder. He will introduce, breeding. So this, for a breeder, you need a very good single line. Simply here, to find out a one line, we need 100, 200. I have to say, this is a mutation. We are not inserting anything. We are simply mutating a gene. This is slightly different from making a transgene. And that is why SDN1 and SDN2, Dr. Sharma has told you, SDN1 and SDN2 has been now deregulated, these plants, by the government of India. This is SDN1. But I'm doing this SDN1. I'm just talking down. I'm not inserting anything. So, but again, thing is that it is not like a rice. It is an asexual propagated plant. I am still having a lot of problem. But sometimes I cannot. It is not going to behave like a rice plant. Where in the next generation you can remove your, or second or third generation you can remove your TGN. Here it is not possible because it's asexual. Maybe we have to take some other strategies. If the proof of concept will we get a good product, if this system is working, then we can think thinking of how to do a completely your tDNA free vector system. So maybe I will that time you will use a your vector free system and go for final. Okay, that is another concept. Proof of concept again that will come up once you get a good product. Whether this CPF system will work or not in the glucose, we still don't know. Right? I don't know. But only I will know after 3 or 4 months. Maybe after 3 or 4 months, my plants will be ready for sequencing. And after that, only after 4 months, if we get some kind of mutation, what kind of mutation, what kind of deletions and all, we have to do a lot of bioinformatic analysis. So after you get the plant, after you get the sequence, you have to do a lot of bioinformatic analysis to find out which complete knockout of the gene or still functioning, or if it is still making a what you call it, partially functional gene. Okay, we don't know what kind of proteins is going to be translated by this gene. So there's a lot of work need to be done once you get the sequence. It is not a 1 year or 2 years. This project will continue up to 10-15 years. We have started for us 3.5 months, 2018, and it will still long way to go. And yes, I need to work on this. Okay. Thank you.
Idea of somebody
For, as I said, in of bean seed is not used as a propagating material. Seed is not used. Naturally, suckers come out. It is an asexual propagated plant. So don't worry about seed to be used as a propagating material. In bean we do not use seed as a propagating material. It is asexually propagated. So that is why whether there is a seed or not does not going to affect their propagation. So ultimately they are going to multiply through sucker, water sucker, sword sucker. That is generally not used for the propagation.
Thank you, sir, for your nice presentation. I hope you understood what CRISPR-Cas system is all about. And how you can do genome modification. See, we are sitting in a very remote place. We are part of Delhi, but we are doing works that are actually international because the BTGP work was international because I'm sure if sir managed to get genome edited lines, I think in the world only 2 groups are working on it. One in Australia, and then Singh is expert in banana genome editing. And there is another group in Nabi, I think.
Nabi.
So this is a very, very good work. I think it will be a very important work. And I hope you have understood everything. Thank you, sir, for your nice presentation once again.
Thank you so much. Thank you all.
I've been looking for an opportunity to get introduced with you all, but I haven't got that opportunity. Yesterday also it was like rush, and all programs started very quickly. Today morning also, my research fellows and all the colleagues, they were busy taking your practical classes. So maybe now, is it possible? All of you can just say a few words about yourself, your name and a few words, and you're from which place? Then you start from. You have to speak a little bit louder.
My name is Bhuvasha. My MC from
Okay.
My name is Manisha Raika and I'm My name is Himama and I'm going to my MC. My name is Peter.
Yours? Yours?